Hi everyone, and thanks so much for joining us. I'm James, Lab Manager here at Cansford Labs, and I'm here with Abbi, who's the Deputy Lab Manager. We both oversee the drug and alcohol testing that plays a part in many of your family law and child protection cases. And we know that these results can carry a lot of weight, and that we feel comes a responsibility to be transparent, clear, and open about how testing works. This webinar is our way of opening up the conversation. We want to give you a better understanding of what happens behind the scenes, clear up any confusion or concerns, and create a space where you feel comfortable asking questions. Whether you're here to learn, raise a concern, or just listen in, we're glad you joined the conversation. If you do have any questions, then feel free to pop these into the Q&A box. So we do have some questions that have been sent in ahead of time. So we just thought perhaps we'd make a start on those. Do you want me to go first, Abbi? Yeah. So the first one that's come through is, can you briefly explain the difference between overview and sectional analysis? So when we do hair analysis, there's two main ways that we can test the hair. And that is either to give an average over perhaps a 3-month period, or break it down into smaller hair sections, which can then provide a smaller window or represent a smaller window of time. So when we talk about overview, that's normally based over a 3-month period. And from a lab perspective, what that means is that 3 centimetres of hair is analysed together, which equates to approximately 3 months. Now that's based on a head hair or scalp hair sample. If we're asked to perform the analysis over a sectional or on a sectional basis, then generally that would indicate that the hair is analysed in 1 centimetre portions, which roughly equate to 1 month in time. Now, you can perform sectional analysis on whatever section length you want, so it doesn't have to be a centimetre. And so, for instance, if we were asked to do a 6-month period, we could base it on overview sections, and that would be 2 3-month hair sections. Or we could do it to sectional, monthly sections, or 2 monthly sections. So it entirely depends on what we're instructed to do, really. I suppose the main difference in terms of what we detect is that an overview section, because it's an integrated average of a longer time period, if there's very low level or sporadic use of a drug, then it's possible for that to be diluted down by periods of non-use. Whereas with section analysis, particularly when smaller sections are analysed, it's more effective at picking up low level or sporadic use. But of course, the section analysis has a cost implication. So that's something to weigh up really, as to whether you're trying to differentiate between potential low level use or more regular chronic use of a drug.
So another question that we've had sent in to us before we started was, why does the Society of Hair Testing recommend an overview rather than segmented testing with alcohol consumption? So to touch on this, the Society of Hair Testing cut-offs that are recommended and are applied at Cansford Laboratories are for 3 or 6 centimetre sections from the root end of the sample, so an overview of either 3 months or 6 months. So the reason for this and why the cut-offs are applied based on overview sections is to do with the way that ETG is incorporated into the hair. As ETG can be reduced by the use of by standard washing procedures, you end up with what's called the washout effect. So as sections get further away from the scalp, levels of ETG can reduce, which means that in certain cases, if somebody is consistently using alcohol, then you can appear to have levels seen as if they're increasing towards the scalp. That doesn't mean that sectional analysis for alcohol testing doesn't have its place. The Society of Hair Testing do recommend that sectional analysis can be beneficial in certain cases, and we do offer sectional analysis here at Cansford Labs. The reason for that is if somebody has drastically changed their alcohol intake, then we actually would be able to see possibly a pattern of reducing levels towards the scalp, which could support this declaration.
Okay, so another question we've had in is regarding what a wash is. So when samples come into the lab, particularly for drug testing, one of the main things we have to try and prevent is that any potential external contamination of that sample being detected. So we wash all samples when they arrive as part of the hair preparation stage. And if there's queries about whether there has been external contamination or whether that person has in fact used or been around someone that's used a drug, we can then go back and analyse that wash solution. There's a scientific consensus of how you interpret those results. So what we do is compare any level of drug or substance within the wash, compare that to what's within the hair, and generally what you would expect is that if someone hasn't been around a drug, that you would detect a significantly higher level of substance within the hair as opposed to on the outside of the hair. Now there's a few kind of complications with that. So things like smoke drugs, so cannabis, heroin, perhaps crack cocaine, that can complicate matters because when someone smokes the drug, their hair actually gets bathed with that smoke or vapour as well. So that can actually complicate matters. But generally, if there's concerns about whether someone has used a drug, then we can tell that by the detection of metabolites or breakdown products. But if that doesn't provide the information we need, then the wash samples can be analysed.
So another question that we've received is when a lab offers a screening or confirmation analysis, what are the differences? So quite often we get asked questions regarding different substances, different medications or things that people have eaten or drank, causing a positive result for something that we've tested for. So in certain cases, certain companies, if they offer screening testing, then it's looking for kind of a wide range of group of substances, and it's possible to get a positive result for a group from the use of something which isn't actually a substance that we've tested for. The testing that we perform here at Cansford Labs is confirmation testing. And that utilises the molecular weight of a substance and the known way in which this will go through our instrumentation and break down to be able to accurately ascribe a drug for what we have detected. So we are able to say and confirm that the substance that we have detected is because of the use of that substance.
Okay, so we've had a question from Andrew Preston, and that is, how do you account for varying hair growth rates for different ethnic groups for drug and alcohol test reporting? So when we talk about variations with different ethnic groups, I know there's a lot of talk at present about kind of racial bias in results. But the main thing that affects particularly levels of substances in hair is the hair colour, not particularly the ethnic background of that person. And particularly when we talk about varying growth rates, it's not down to necessarily the person's ethnic group, it's down to the hair type. So there's three main hair types, and they're Caucasian, Afro-Caribbean, and Asian. And each of those have distinct growth rates. They also have different kind of cross-sectional diameters, and also mainly different hair colours. So when you think of Afro-Caribbean and Asian, most of those hair types are darker in colour, and then you have the Caucasian, which can be a mix from unpigmented or white through to very dark brown or black. So we take the same approach as kind of most other hair testing labs, and that is based on Society of Hair Testing SOHD guidance, and they state that an approximate 1 centimetre per month hair growth rate should be applied to provide different time periods. Now, all labs accept that there is a range of hair growth rates, and generally that's accepted is ranging from 0.7 centimetres per month up to 1.5 centimetres per month. So when we and other hair testing labs provide results, they will make quite clear that there's an approximate 1 centimetre per month growth rate being applied. And that's not saying that that particular donor's hair does grow 1 centimetre per month, but that's kind of an assumed average. So I don't know if you require any more information on that, Andrew. If so, pop it into the Q&A box. But we've had some other questions come in.
So we have a question here from Anna Miguel. What happens when the individual has used drugs but also lives in an environment where drugs are being used around them? There'll be drug incorporated but also a relatively high concentration coming from the wash. How would you interpret it? So in this case, we don't routinely analyse wash samples in cases where drug use has already been supported through the analysis of the hair sample. So in order for us to say that a result is consistent with use, for example with cocaine, we would expect to detect the metabolite of cocaine, which is benzalkonium, which is the main metabolite, at a certain level in comparison to the level of the cocaine in the sample, and we would be able to say that that result is consistent with use. In cases where the wash has then been requested as well because of possible external contamination issues, then we would say that it is likely that the result is due to use of the drug, because we've confirmed it with that metabolite as well, and being in an environment where that substance is present too. We would only do a wash from the beginning point in instances where we're not able to confirm use with that metabolite, and we need to analyse that wash to provide further information.
Okay, so I think we're just waiting for some more Q&As to come through, and we have got the other questions that were provided ahead of time. So the next one of those is regarding how do ethanol containing hair products influence results? So when we talk about hair analysis for alcohol markers, there are two main markers that can be done. That's ETG and ETPA. The ETG, Abbi touched on previously when she was talking about the section or non-section for alcohol analysis. ETPA is the main marker that can be affected by hair products that contain ethanol, and that's because ETPA can actually be formed in the presence of ethanol. So if someone does use a hair product containing something like such as hairspray, hair gels, things like that contain ethanol, then that should be declared at the point of collection, because that can then be factored into the interpretation. And so it is quite important because ETPA levels can be elevated. And so generally in those sorts of situations, if ETG and possibly a blood marker have also been done, then everything can be interpreted together. ETG isn't affected by hair products that contain ethanol, but it can be affected by chemical hair treatments. So that's why the general recommendation when there's been chemical hair treatment is for 2 hair alcohol markers to be done, and also blood alongside that. Do you want me to link into the next one on the list, and then we've got some more coming through, I think. If we provide lots of detail on the environment and circumstances around a client, will that impact how the experts interpret the results? Well, I suppose the blunt answer really is no, it shouldn't, because all an expert is doing is interpreting the findings of the analytical results. Now, the danger is that if lots of contextual information or information which could sway that person's view is provided, then there's a danger that the interpretation could be given one way or another. And so at Cansford Labs, we report the findings of the analysis that we perform in-house. That's the first part. If then we're given other information regarding perhaps being exposed to drugs on a frequent basis, then we would provide secondary kind of interpretation of that. But the overall interpretation shouldn't be really affected, because if you find a substance within a hair sample, then that would indicate that perhaps that person has used or come into contact with the drug. So do we want to go back to some of these that have come in?
So we've had a question in from Emma Brady, and that question is, what effects does colouring and bleaching have on the sample? So levels of drugs and alcohol markers can be reduced by the use of chemical treatments that damage the hair, such as hair dye, hair bleaching, perming, anything that can cause the hair follicle to be damaged. When we do report on results and we provide that expert interpretation, we do take into consideration whether the hair has been chemically treated. When we collect the sample, this is asked of the donor at the time of collection as to when the hair was last treated. We also will make a note of the chemical treatment, whether there appears to be any dye or any dye line when the sample is received at the lab before sample preparation. And this does mean that levels can be reduced, but this is always going to be incorporated into the report.
We've got a follow-up question from Anna McGill about the finding of benzoylecgonine on the outside of the hair. Do you find that issue too? So I suppose the main thing, I don't know what your background is, Anna, but one of the main things with finding benzoylecgonine on the outside of the hair, that can indicate mainly in child cases. So if we analyse perhaps samples from young children who have frequent contact with caregivers, if there's poor hygiene, then we do, or you could potentially find things like ETG and also benzoylecgonine on the outside of the hair. And that's mainly down to poor hygiene practices. So in urine, following cocaine usage, you tend to find quite high levels of benzoylecgonine in the urine. And also following alcohol ingestion, you find quite high levels of ETG. So there's a possibility of finding those on the outside of the hair. We presented some findings from kind of research that we did, I don't know, probably 10, 12 years ago, saying about how benzoylecgonine and other cocaine metabolites can be found at very low levels in cocaine. And that's mainly down to kind of degradation of the cocaine, and also can be influenced by how the cocaine is brought into the country. So perhaps if it's smuggled in bottles of alcohol, then you can find low levels of cacophylline in that cocaine as well. But generally the levels at which you find them, or the metabolites, are so low that they wouldn't generally affect the overall interpretation of the analysis.
So we've got another question that was sent in previously, which we will touch on. So that is, how long does someone need to be using drugs or alcohol for it to show up in a test? And if they stopped 3 months ago after using heavily for years, would it still be detected? So to split this up, how long does someone need to be using drugs or alcohol for it to show up in a test? So this all depends on the frequency of use and the kind of the purity of use. So the cut-offs that we apply here at Cansford Labs are recommended by the Society of Hair Testing. And the reason for these cut-offs and the reason that these are used is to rule out detection of substances from possible single-time use or historical use of a drug or unintentional use of a drug providing a positive result. So if somebody has used a drug on kind of a one-off occasion, we wouldn't necessarily expect this to be detected above that cut-off. If somebody is using drugs more frequently, then we would be expecting to detect those within the section that we're looking for. So if we were looking at a 1 centimetre section, then that would cover approximately 1 month history, and 3 centimetres would cover that approximately 3 months of history. In terms of if they were previously using heavily and would it still be detected 3 months after ceased usage, that is possible. And the reason for that is to do with the way that hair grows. so the hair growth rate. So the way that hair grows is that it's not all growing consistently at one time. You have some hairs which are in what we call the resting phase. So these hairs are not actively growing, but they can contain traces of drugs from previous use. So when somebody stops using a drug, what we would expect is to see decreasing levels of the substance towards the scalp. And that can last for a few months after somebody has stopped using a drug. But this would all be explained and discussed in the interpretation of the expert witness statement.
Okay, so we've got a question from Mohan. Does every tester use 1 centimetre hair growth rate to represent a month's worth of testing? I would say that probably most labs within the UK do. I'm aware that one lab doesn't. They apply a hair growth rate to 1.2 centimetres per month. I'm not quite sure the scientific basis of this. I haven't really read any peer-reviewed articles to suggest a hair growth rate of 1.2 centimetres per month. It may be that perhaps their experience is more from North America, because I know that they apply a growth rate in inches, so perhaps the conversion into centimetres has led to some confusion there. But in terms of the 1 centimetre per month growth rate, that is something that is stated by the Society of Hair Testing. I know there's some information going around at present about the guidelines are very outdated. You know, in my career, I've been through a number of reviews of the consensus, and there have been a number of updates over the years. So yeah, the guidelines aren't necessarily, you know, been updated in the most recent year, but certainly in the last couple of years, and no doubt they'll be reviewed within the next 12 months as well. And I suppose kind of the main thing with the Society of Hair Testing is that, you know, it's from a wide range of experts. So those experts are from across the globe. They meet kind of every other year at their conference. And if there are any kind of major findings, then those are generally discussed and raised. And if there was any kind of major failing in the guidelines, then I have no doubt that those guidelines would have been updated.
So we've had another question come through from Andrew Preston, and this is another testing provider I've been speaking to says that the Society of Hair Testing head hair cutoff values can't be trusted for sample donors from non-Caucasian ethnic groups with drug testing because, like you say, all testing labs have to use the average growth rate of 1 centimetre per month. What is your opinion on this, please? So in regards to the 1 centimetre per month, we've touched on that a few times now. So most labs, as James said, do base on the 1 centimetre per month as recommended by the Society of Hair Testing. However, we all are aware that growth rates can vary between individuals. So based on this is based on an average. So what we need to all be aware of is this is for guidance only. So the time periods that we provide are approximates, and this can vary between individuals dependent on what their exact growth rate is. So when we have samples from non-Caucasian ethnic groups, we will comment on the fact that the growth rate may vary, and it may actually cover a longer time period than what is stated. And this is due to the way in which this hair grows. There may be more hair in the resting phase in the sample that we're testing. So it's just important for people to remember that the time periods that we are providing are approximates, and the sample needs to be looked at as a whole, along with other evidence as well.
Yeah, and I think, as Abbi has just said, going on from that, Andrew's put that the SOHD head hair cut-off values can't be trusted to sample donors from non-Caucasian ethnic groups. Well, as I mentioned earlier, the hair type is one thing. So the main thing that affects the levels in those hair types, different hair types, is the hair colour. So you could have a Caucasian individual with an Afro-Caribbean hair type, and that could have the same characteristics as non-Caucasian with Afro-Caribbean. So we can't just talk about the donors being non-Caucasian or Caucasian. It's mainly down to the hair type and colour. So we then have a question from James Doak. Does accuracy or time frame differ for body hair samples? So I suppose it's a two-part question really, because the accuracy is the ability to detect a substance within that hair sample. So the accuracy is the same for head hair and body hair. The main factor is, as you've kind of touched on, James, is the time frame. So because body hair, we can't restrict it to a specific time period, there is a chance that if that body hair is longer and that person has perhaps historically used or only used very sporadically, that because it's tested as a whole section or overview or integrated average, then there's a chance for any low levels to be diluted by periods of non-use. So I think if someone is having drug or alcohol testing done, head hair is always the preferred choice, because you can restrict the time period to as long or short as you want. The proportion of resting phase, which Abbi touched on, is much lower in head hair, so roughly up to 10%, whereas body hair can be up to about 50%. So that means that body hair can cover a much longer time period. So yeah, body hair is more complicated in kind of indicating timeframes of usage.
So we've got another question from Leonie Watson. Is it possible for markers of crack cocaine to be found in a sample just from cocaine use and not crack cocaine use? So this is something that has been brought up recently. There has been a bit of kind of talk about this. So yes, it is possible to have the marker for crack cocaine be found in a sample. But the reason for that is if somebody has possibly used thermal treatments on their hair, so the use of straighteners. If cocaine is present in the hair sample, it's possible for the crack cocaine marker to be produced, as this is produced through the use of heat. And it's also possible for the levels of benzoylecgonine to be increased as well. This is something that we are aware of, and this is something that has been discussed in expert literature and will be commented upon within the report if needed. However, the interpretation of results will be down to the levels of that marker that is detected. So if we are getting low levels of that crack cocaine marker and we have the results are consistent with cocaine use, then it will be commented upon that it is possible that it may be due to crack cocaine use. However, if thermal treatments have been used, that may have resulted in that. If we're detecting higher levels of the crack cocaine marker, then that's more indicative of crack cocaine use itself.
Okay, so the next question we've received ahead of time. Is it possible to have tested above the cutoff level when you've only shared a bed or been in the vicinity of a drug user? So really, I suppose that if it's kind of on an occasional basis, then you wouldn't expect for exposure to a drug to, result in the detection in a hair sample. The question is really about the vicinity of a drug user, because if that is on a very frequent basis and potentially something like crack cocaine, then it could there could be some detection. I suppose it boils down to whether cutoffs have been used or not. One of the suppose benefits of a cutoff is to try and prevent unintentional use or exposure, ingestion or exposure to a drug resulting in the positive detection in a hair sample. So you would expect that if there was periodic exposure to a drug, then it wouldn't really show up. The problem is when cutoffs aren't used. There's been quite widely documented cases in peer-reviewed journals looking at things like vets, certain professions being exposed occupationally to different drugs. So in the case of vets, it was ketamine. And I think we, amongst some other kind of hair testing labs, share the same view that if cutoffs aren't going to be used, then it's going to lead to a result. It's going to lead to a rise in the detection of findings or positive findings in hair samples, and that isn't necessarily down to active use of a drug. It could be occupationally, it could be personally being exposed. So that there's a lot to consider there. But generally, you wouldn't expect kind of sharing a bed or vicinity of drug user on a kind of sporadic basis to cause positive findings.
So we've had a question in from Kate Jones. What is the impact of binge drinking on alcohol testing? So we'll break this down into the different types of alcohol testing. So firstly, the World Health Organization defines excessive drinking, chronic excessive drinking, as the consumption of 60 grams of ethanol per day over a period of many months. So that equates to approximately 7.5 units of alcohol per day. So when we're looking at alcohol testing in hair samples, so for ETG and fatty acids, ETPA, if we're basing the cutoffs for chronic excessive alcohol consumption, it is possible that if somebody is binge drinking, so drinking to excess on, say, certain days of the week, so maybe over a weekend, or for somebody that is consistently drinking throughout the week to produce a similar level of ETG. So we're not able to differentiate between somebody that has consumed excessively for part of the week or just consumed consistently throughout the week. It's down to the amount of alcohol that's consumed within that approximate time period that produces a result, and that is then based upon the cutoffs which we apply. So at Cansford Labs, we're able to differentiate between irregular or abstinence from alcohol, a level detected below 5 picograms per milligram, or a value above 30 is indicative of chronic excessive alcohol consumption. So following on from that, the other testing that we can do for alcohol markers is the PEth test, which is a blood test. So at Cansford, we do this via a finger prick, a dry blood spot test. And if somebody has been binge drinking and then has a PEth test, it is possible that if the test is done possibly the day or two after that episode of binge drinking, that a higher level of PEth may be produced, because drinking 5 drinks in one sitting will produce a higher level of PEth than consuming that over a period of time. However, that PEth test is looking at a 1 month history of alcohol consumption. So combining the PEth blood alcohol test along with the hair alcohol markers is able to provide a better indication of somebody's alcohol consumption over a period of time. If somebody's been consuming to a chronic excessive level over the previous 3 to 6 months, which is indicated by a hair test, a PEth blood test may show that they have moderated or reduced their alcohol consumption in that 1 month period prior to testing.
Okay, so we've had a question from Leonie Watson. Is the best way to consider binge or social drinking via Strand bracelet? I suppose it depends on kind of how much information is required, because as Abbi just touched on, PEth is routinely used for determining whether someone is regularly consuming alcohol or not. Strand bracelets have their uses, but it's quite an invasive way of monitoring alcohol consumption. So, it has to be worn 24/7 by that person for a period of months. They can't just be removed when that person doesn't require monitoring. In terms of alternatives to Scram, I would say that there are alternatives. A PEth would be something we would generally recommend. There's other newer products on the market. We don't yet offer them, but things like breathalyzers with facial recognition, they can be quite useful for cases where perhaps the parent only has custody of a child perhaps for a day every 2 weeks, because that person only has to be monitored for perhaps a day or so, whereas Scram would have to be worn all of the time. So I wouldn't say that Scram is probably the best way of assessing binge or social drinking. PEF could be used, but that would be more likely used to indicate whether there has been regular consumption of alcohol or not.
So we have had a question from Sophia. Does herbal tea, yerba mate tea, affect testing results for cannabis and cocaine? I believe we have come across this question before, and we can't find evidence that yerba mate tea contains cocaine or cannabis. The plant contains high caffeine, and therefore, as we discussed earlier, the testing that we perform at Cansford Labs is confirmation testing, and it would not provide a false positive result for cocaine.
So we've also had a question from Marie Nelson. Please can I ask what you would be able to offer if a customer requested a segmented test report on a different growth rate, for example, 0.7 centimetres per month? Well, in terms of kind of providing results, we can easily provide the approximate periods covered by the hair sections analysed if the hair growth rate was 0.7 centimetres per month. So quite often we're asked this sort of question, and then we can easily provide the time periods covered by the sections, assuming growth rates of 0.7, 1 and 1.5 centimetres per month. So that would probably be the easiest way around that, I would say. So we would still analyse the sample as 1 centimetre hair sections, but then we would calculate the time periods assuming a slower growth rate.
So another question that we've received prior to this was, could you explain your 3-day turnaround time? Does that include the expert report as well? So yes, that does include the expert report as well. So what we guarantee is that sample results, including that expert report, will be received by 5 p.m. on day 3 that the sample has been received at the lab. So when those samples have been collected and posted, once they're received at the lab, that is classed as day 1. So the samples will be prepared on that day and produced through the lab and onto the instrumentation. Usually results will then be processed and looked at the following day, which is day 2. And in the majority of cases, results will be issued on day 2, which is including that expert report. However, the guarantee is within 3 working days.
So another question we've had was, what do we need to look out for when choosing a lab to test with in regard to accreditation? ERA accredited, what does it mean? So for performing laboratory analysis, organisations should be accredited to the relevant international standard, and for testing labs that's ISO 17025. That's currently within the UK, awarded by UCAS, the UK accreditation body. Now some organisations will have accreditation for ISO 9001, but that only looks at one portion of the company, so that just looks at the quality management system. It doesn't ensure the accuracy or integrity of the results. Whereas ISO 17025, which us and other companies like ourselves are accredited to, is kind of an in-depth analysis by UCAS to make sure that all the systems in place are suitable. You have adequate resourcing in terms of people as well as instrumentation. It also makes sure that you're compliant with all current industry guidance, and also that kind of your testing is accurate on an ongoing basis. So it ensures that you're a competent organisation. All information on this can be found on UCAS website. I think we probably have a blog about accreditation too that we can probably send links to. But the main thing is whether an organisation is accredited to ISO 17025 or whether it's just the organisation that's accredited to ISO 9001.
Okay. So we have a question from Andrew Preston. Do you offer head-hair mentally segmented alcohol testing for both ETG and FAE, please? So yes, we do. As mentioned earlier, at Cansford Labs, we are able to offer segmented analysis for ETG and ETPA, which is the fatty acid ethyl ester marker that we test for. We have been doing segmented analysis for alcohol testing for many years. And we have a lot of data and research surrounding this and surrounding the benefits of it. We have got expertise within publishing this information as well and presenting it at conferences, and we are able to provide an interpretation of these results and what they mean in comparison to what other companies may offer in terms of overviews and how the results will fit within the Society of Hair Testing recommendations.
So we've got about 10 minutes left, so if you do have any questions, then feel free to put those through. We will be sending out a recording and also a copy of all the Q&As in the next few days. But if you do have any, then feel free to send those through. So another question we've been asked is, there are news articles circulating that suggest that hair type, particularly darker hair, may retain substances differently, potentially affecting test results. Can you explain this? So yeah, there has been a fair bit of publicity, for the past 6 to 12 months, I suppose, indicating this. It's nothing new in the industry, really. It's been known about for a number of years. I know there was a study done, must be over 20 years ago, but it was quite a small study, only about 20 participants, and it only looked at one drug, I think codeine. So it is quite an outdated study, but it's something that we should consider. Now, as we've both touched on throughout today's session, hair colour can have an effect, and generally the effect is, for some substances, is that darker colour hair can retain higher levels of substances. So things like cocaine is probably one of the most affected. Now, not all substances are retained or incorporated at such a high level as cocaine is in darker colour hair. So you do get variations among different substances. So that's one of the main things to consider, really. So, but what I would say is that all samples, so when we and other labs compare samples to other positive samples, that data set is from a wide variety of data. And when you think that the vast majority of the population have darker colour hair, or brown and darker colour, those results are going to reflect that kind of proportion of the population having darker colour hair. So yes, darker colour hair can contain higher levels of substances, mainly drugs, but there are variations within those substances. It's something which is continually being updated and researched, because the thinking behind it originally was that melanin was the primary way that drugs are retained in hair. Now, we know that's not true, because if you have someone with white or unpigmented hair, they can still have positive drug results. So it's not solely down to melanin. There's other ways of incorporation, sweat, and other perhaps ways such as that, sebum. So we would always kind of touch upon if there is a potential impact on findings. And I think most other labs like ourselves would have the same view, really. Darker colour hair can have higher test results for some drugs, but it's something that's been considered for the past 10-15 years. It's certainly nothing new.
So another question that we've had previously is, why do cut-offs exist? Who do they protect? So as we've touched on a few times throughout this session, the cut-offs that we apply at Cansford are based on the Society of Hair Testing recommendations. So cut-offs are applied, and in our case, the reason that we use these cut-offs and a lot of other organisations such as ourselves use these cut-offs is to standardise almost across the industry, so that it is given a kind of a fair representation across the industry when people are having these tests done. The reason for them is, as mentioned previously, is to rule out the possibility of getting a positive result from single-time use of a drug, unexpected, unexplained use of a drug, unintentional, and from environmental exposure, along with historic use of a drug, and as James mentioned, through certain workplaces, possible exposure through that way. And the reason for this is to avoid providing these positive results, along with maybe a misinterpretation of these results, and saying that somebody is using a substance that they are not using. They protect the individuals from having these possible low-level results reported as use, when there could be other reasons for those substances to be there. There are reasons why cut-offs are not necessarily applied, and these again are recommended by the Society of Hair Testing. So in such as things as children cases or drug facilitated crime cases, where it's imperative to see whether or not there is a substance there, as opposed to whether or not it's just detected above the cut-off level, because anything below the cut-off level in those cases is important to see, as they shouldn't be present.
Okay, so I think that probably brings a close to today's session. If you do have any questions, then feel free to drop us an email. We're keen to make sure that everything out there is correct, any information out there is kind of reflective of current scientific practices and is sound from a scientific perspective. We share the views of many other testing labs. We have good relationships with most of those in the UK. So, if you do have any questions, just drop us an email and we'll be able to, help out and try and clear things up for you. So thank you for your time today, and hopefully speak to you soon.
Thank you.
Thank you.